I'm here today to share more about precision fermentation done at Allozymes for the production of high value chemicals. Compared to an enzymatic reaction with a single enzyme, precision fermentation is complex and involves multiple enzymes to perform a series of cascade reactions. The cascade reaction starts with a simple substrate as a sugar and flows through the synthetic pathway and gets transformed into a product of interest by a series of enzymatic reactions. This complex reaction is possible as it happens inside the microbial cells, and we call these engineered microbes as microbial cell factories. Fermentation, the traditional method of producing products like bread, wine, and beer, uses natural microbes, whereas precision fermentation leverages cutting edge biotechnology to engineer these microbial cell factories to produce a wide range of valuable products. It can be from biofuels to pharmaceuticals all the way to food ingredients. By carefully manipulating the genetic makeup of these microbes, scientists can create strains that efficiently produce desired compounds. And some exciting applications of precision fermentation include producing sustainable alternatives to animal derived or plant derived products and developing new drugs and vaccines. Why do we need microbes for executing this complex reaction? As shown on the left, microbes naturally converts sugar, such as glucose, to energy-rich precursors while performing thousands of enzymatic reactions for its own metabolism. By tapping into these precursors for our target production, we are leveraging the efficiency of naturally involved pathways while using simple and sustainable starting materials. As shown on the right side, enzymes require different cofactors such as ATP, NADH, NADPH as energy sources for completing a biocatalytic reaction most of the times. And microbes generate and provide these cofactors for executing our own target reactions. And these cofactors are also continuously recycled with the metabolic pathways of the microbes. This slide shows the full overview of a precision fermentation happening in allozymes. First, to convert the sugar feedstock all the way to pure high quality product, interdisciplinary research and development is required. Allozymes have these R&D expertise in-house. Let me explain step by step. After choosing the feedstock, strain engineering is initiated. It involves genetic engineering to add or remove enzymes or to tune expression levels of the newly added enzymes. And then enzyme engineering is required to develop a better enzyme for the process. Then comes in metabolic engineering that is done to effectively incorporate the synthetic pathway with the naturally occurring metabolism inside the microbe. During this whole process, bioinformatics provides the in silico support and biochemical characterization provides the analytical methods to understand the process. Finally, there is omics, which gives us a detailed picture in different levels, such as transcriptome, proteome, and metabolome. So, all these different fields come in together to develop a perfect strain. But it doesn't happen overnight. It needs to go through a few rounds of development. And the developed strain undergoes process development, which is the next stage. Strain engineering and process development work hand in hand in allozymes and undergoes a few cycles of DBTL, which is design, build, test, and learn to develop an efficient microbial factory. This microbial cell factory, which is validated in our lab, is then given for scale up in different levels, and finally, a pure product is commercialized. Though there are several synbio players in the market doing precision fermentation, the upper hand of allozymes is its enzyme targeted engineering. Let me explain how. Almost all the process, as shown on the left, almost all the process that go from idea to commercialization have to cross this steep called valley of death here. So this is where you have limited resources to develop a process that is commercially viable. To increase the commercial viability, it is crucial to reach the targets in three very important performance metrics, which is shown as here: TRI, titer, rate, and yield. By quickly achieving the targets performance metrics of TRI, you can cross the valley of death faster and significantly increase the chances of commercial success. As shown on the right, there are four promising engineering steps that have been well studied for several years to increase the TRI, which are: first is increasing the precursor and cofactor supply, second is eliminating bottleneck enzymes or steps in the pathway, third is improving the enzyme specificity to not produce any byproducts and also pull your product flex completely towards your product. And fourth is downregulating competing enzymes or pathways not to waste your carbon. All of these steps require some forms of enzyme discovery and enzyme engineering to bring up the efficiency. And by doing engineering, you are significantly increase the cap of maximum production in this process, thus quickly achieving the tighter rate and yield. So the bottom line here is efficient enzymes are the key to increase tighter rate and yield, and Allozymes is doing that using its enzyme targeted engineering. Here I have a case study where we have developed a precision fermentation process for sesquiterpenoid production using engineered E. coli with Allozymes expertise. So sesquiterpenoid is a bioactive chemical which is derived from FPP that have anti-inflammatory, antioxidant, and several other valuable properties. Currently, these molecules are mainly extracted from plants, which is not sustainable. So we have developed a microbial platform for producing this sesquiterpenoid in large scale. For that, we have first assembled a hybrid pathway in E. coli involving 15 to 20 enzymes. And during the process, we have discovered, assembled, and expressed several non native enzymes into the E. coli. Then we identified the bottleneck in the pathway. We engineered the three key enzymes in the pathway to increase titer, which is one of the key metrics. Then we developed an optimized bioprocess to increase rate, which is another very important metric. Further, we balance the pathway flux because the cell needs to grow while producing this sesquiterpenoid. So you have to balance the pathway flux. You don't want to completely kill the cell, and you don't want the cells to just grow without doing any of your production. So you have to balance the pathway flux using metabolic engineering. Thereby, we have increased the yield of this process, which is the third metric. Finally, we have a downstream process developed in-house, and with that, we have isolated the product with high purity and made this process ready for commercialization.